Gregor Mendel, through his work on pea plants, discovered the fundamental laws of inheritance. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive alleles. He recognized the mathematical patterns of inheritance from one generation to the next. Mendel's laws of heredity are usually stated as the law of segregation. Each inherited trait is defined by a gene pair, the law of independent assortment. Genes for different traits are sorted separately from one another so that the inheritance of one trait is not dependent on the other, and the law of dominance. An organism with alternate forms of a gene will express the form that is dominant. These genetic experiments Mendel did with pea plants took him 8 years, and he published his results in 1865.
Hi, DNA or RNA. DNA is a genomic material in cells that contains the genetic information used in the development and functioning of all known living organisms. DNA, along with RNA and proteins, is one of the three major macromolecules that are essential for life. Most of DNA is located in the nucleus, although a small amount can be found in mitochondria. Within the nucleus of eukaryotic cells, DNA is organized into structures called chromosomes. The complete set of chromosomes in a cell makes up a genome. The human genome has approximately 3 billion base pairs of DNA arranged into 46 chromosomes. The information carried by DNA is held in sequence of pieces of DNA called genes.
Punnett squares use mathematical probability to help predict the genotype and phenotype combinations in genetic process. The first step is to identify the genotypes that are being crossed. The second step is to draw a Punnett square. The third step is to label the Punnett square. The fourth step is to determine possible offspring by filling in the Punnett square.
The step number 5 is to determine the possible phenotypes of the offspring. Step number 6 is answer questions asked in the problem.
I'm going to talk about mutations, and mutations are heritable changes in genetic information and are some gene mutations that, for example, is substitutions, insertions, and deletions. It's important to know that genetic material can be altered by natural events or by artificial events, and the resulting mutations may or may not affect the organism. And also, many mutations are produced by errors in genetic process. Well, also there are harmful and helpful mutations. The effects of mutations on genes vary, so some have little or no effect, and some produce a lot of beneficial variations, and others negatively disrupt genetic function.
Meiosis is a process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Meiosis has 4 phases. The first one, prophase, that is, each replicated chromosome pairs with its corresponding homologous chromosome. Then, during metaphase of meiosis, paired homologous chromosomes line up across the center of the cell, and during anaphase, spindle fibers pull each homologous chromosomes paired toward opposite ends of the cell. The next phase is telophase, in which a nuclear membrane forms around each cluster of chromosomes, and the last one in cytokinesis follows telophase, that is, forming 2 new cells. In meiosis 2, the 2 cells now enter a second meiotic division. Unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis 2. It has 4 phases, and the first one is prophase. As the cell enters prophase 2, their chromosomes, each consisting of 2 chromatids, become visible. Then it's metaphase 2, anaphase 2, telophase 2, and cytokinesis. During metaphase, chromosomes line up in the center of each cell. The final 4 phases of meiosis 2 are similar to those in meiosis 1. However, the result is 4 haploid daughter cells.
En conclusión, hay elementos muy importantes a la hora de hablar de negocios. Tu expresión oral es una de las principales. Si no tienes una pequeña conexión con la persona con la que estás hablando, será muy difícil que entienda lo que estás tratando de comunicar. Todos estos elementos te ayudan a tener un mayor entendimiento del lado del cliente o del lado del comprador, haciendo del área empresarial un campo de entendimiento más eficaz y seguro. Sin estos elementos, las empresas no tendrían negocios, ya que los negocios no podrían tener sentido o no serían de la manera correcta.